So here are some differences between the eukaryotic genome and prokaryotic genome. Eukaryotes, typically our genetic material, is going to be formatted as a double helix with multiple pieces. We're also going to have multiple chromosomes, and our genomes are going to be comparatively larger to prokaryotes. Transcription and translation are going to be physically separate because we are going to transcribe in the nucleus and then eventually send that transcript out of the nucleus into the cytoplasm where it's eventually translated. So physically separated by that nuclear envelope. And then each gene is going to typically have its own promoter if you're a eukaryote. And our genes are going to be located fairly far apart. And we have three different types of RNA polymerases whose binding will depend on other proteins. Now, when we're looking at prokaryotes, they're typically going to have circular DNA, and oftentimes they're going to have a single chromosome, so one single main chromosome with many possible plasmids or smaller circular DNA. Their genomes are going to be comparatively smaller to eukaryotes, smaller than eukaryotes, and their transcription and translation is generally not separated because they don't have a nucleus. And so usually as they're transcribing genes, the ribosomes are going to come along and translate it as it's being pumped out. Oftentimes prokaryotes will have one promoter, as we saw earlier, one promoter for several genes, several functional genes. Usually those genes are going to be related to one another in function. And you're also going to have their genes, or prokaryotes' genes, are going to be clustered together. And typically one RNA polymerase is going to be used and is not going to depend on other proteins for binding. So let's look at eukaryotic transcription. So eukaryotic transcription, typically we're going to have some kind of promoter sequence. In eukaryotes, we call this the Tata box because it's a series of T's and A's and T's and A's. So it's a Tata box where the double strand will open and transcription factors will bind. So here, looking at the first part A, you have transcription factor 2D, which is a Tata box binding protein. So that's going to bind to the Tata box, and then from here we get this cascade. You get transcription factor 2A, transcription factor 2B binding, which then recruit transcription factor 2E, 2H, and then 2F, which is bound to RNA polymerase. And eventually you form the full complex needed in order to progress with transcription. And then in addition to this, it's going to cost a lot of energy. You might use many various forms of energy, and eventually that will set transcription underway. So we're not going to have to remember all of these different transcription factors in the order that they're binding. Know that transcription factors are going to be heavily used in allowing this RNA polymerase to bind. So here it says transcription factor 2 and other transcription factors form the transcription initiation site. And then after the transcription factors have arranged, the DNA changes shape, allowing RNA polymerase 2 to bind. Now we also have some other things that we will notice on eukaryotic genomes is we have things called enhancers. So enhancers are going to be a stretch of DNA that a activator can bind to. So enhancers are upstream or downstream, so they can be, upstream, so before the gene of interest, or downstream from the gene of interest. And when they're bound to by activators, they will increase the rate of transcription. So typically what happens is that activator will bind to the enhancer stretch, and that will cause DNA to fold in a way or recruit proteins, allowing it to fold in a way to enhance transcription. Now, the opposite of enhancers, we have things called silencers, which could be upstream or downstream sequences that, when bound to by repressors, will do the inverse. It'll repress transcription. It'll prevent transcription from happening. And oftentimes, these are going to play a role in cell differentiation. So we have enhancers, which are stretches of DNA bound to by activators, and we have silencers, which are stretches of DNA bound to by repressors. And the names are kind of self-explanatory. Enhancers bound to by activators will enhance or activate transcription, and then silencers will silence transcription when bound to by repressors, which repress transcription. Okay, now eukaryotic organisms have a lot of different types of post-transcriptional processing, a lot of different ways we can modify our gene expression after we've already created that transcript, after we've created that messenger RNA. So one way is you can actually get alternative splicing of the mRNA, meaning you can take some exons out of the full protein, or you could leave some in. So you can splice it in alternative ways to yield different proteins. So you can splice it one way, or you could splice a different way. I'll show you a picture of that in a little bit. Next, we have the end cap of mature mRNA, which may or may not be modified properly to begin transcription. So we have a few things that we do after we've created our messenger mRNA. We add a cap at one end and a poly A tail to the other end, which I'll show you a picture of. And those kind of serve as signaling molecules to the nucleus that you can bring that out of the nucleus into the cytoplasm. And then we have gene silencing by what we would call microRNA or small interfering RNA. We could actually create little chunks of RNA that are with the intent of having them bind to the mature mRNA that we've created. And then lastly, we have eventually when we've created that protein, when the ribosomes translated that messenger RNA into a protein, we can actually just simply break down that protein via hydrolysis. So generally, prokaryotes are not going to regulate transcription in these ways. Typically what they do is they'll tend to alter their ribosomes to either turn off translation or turn on translation. Okay, so here we have the alternative splicing that we're going to see in eukaryotes. So if you look here, you have the original strand of DNA, which we transcribe into the messenger RNA. And as we know, we will splice out, or we will splice exons together by pulling out the introns. But the thing is, you have exon 1, 2, 3, and 4, and 5. Now here is an example of if we left all of these exons in, we spliced them all together, sent it out of the nucleus, we would create one type of protein. However, let's say we omit exon 3. We treat this as though it's an intron, we cut it out, and then we splice exon 1, 2, 4, and 5 together, and we omit exon 3. We're going to get a different type of protein because it's going to have a different sequence of nucleotides, which eventually leads to a different protein. And then in this case, you have 1, 2, 3, and 5, so you're omitting exon 4. Again, you're getting another type of protein. So you have alternative splicing as a way of post-processing or post-transcription regulation of gene expression. Now, like I was mentioning earlier, you have a G-cap, which you'll add to the top of, or the 5' end actually, specifically. The 5' end of your mRNA is actually makes a 5' to 5' bond. So you'll have this G-cap at one end, and then you'll create a poly A tail, poly meaning many, A for adenine, and a tail at one end. And so this poly A tail helps with stability of your mature mRNA as well as your G-cap. And then in addition to those qualities, in addition to adding stability to your mature mRNA, these things also aid in the transport of your messenger RNA outside of the nucleus into the cytoplasm. These are just the enzymes that actually catalyze the process of creating that 5' cap, that G cap, and the poly A tail. So guanylyl transferase, so it ends in an ase, it's an enzyme. Poly A polymerase, you're making a polymer of A, right? You're making a poly A tail, and it's an ase because it's an enzyme. All right, next we have microRNA, which are very We could talk about this for a really long time, but in short, we actually have most organisms are able to identify double-stranded RNA. And the reason for that is because a lot of viruses, they'll actually, their genomes are in the form of double-stranded mRNA, or double-stranded RNA, sorry. So our viral immunity, we actually have machinery built around detecting double-stranded RNA and breaking down double-stranded RNA. And so what we could do here is, let's say we've created too much of a transcript that we don't necessarily need, or we want to make sure that that transcript isn't overexpressed. It is not picked up by too many ribosomes and translated into too many proteins. One thing we might do is we'll create something called microRNA in our nucleus, as though we would be creating the messenger RNA, and then we allow it to be modified by an enzyme called dicer. So what it's going to do is we create this RNA. It usually folds onto itself, and then this enzyme called dicer will cut that loop. So it's cut this loop off, giving us two strands of RNA that are complementary to each other. Now, eventually, one of these will dislodge and find its corresponding messenger RNA in the cytoplasm. So usually in plant cells, the microRNA is usually perfectly complementary to the target mRNA molecule. So the microRNA will bond with it and then cause the mRNA to eventually break down. Now, in animal cells, typically it's not a one-to-one, it's not a perfect fit, but it's good enough to create double-stranded RNA. And so if you look here, you have this ribosome. Well, ribosomes will read single-stranded RNA. They're not going to read double-stranded RNA, so it's going to impede the progression of this ribosome in its translation process. Now, in addition to that, it's putting a stall. So it's putting a stall on this messenger RNA, and over time, what's going to happen is your poly A tail will eventually degrade, and then eventually will lead to the breakdown of the messenger RNA. Now, lastly, I think this is lastly, we're talking about breaking down the protein. So once we've created the transcript, sent it out of the nucleus, and it's picked up by a ribosome, that ribosome translates that transcript into a protein. Well, let's say we want to regulate the amount of protein we have present, we could actually just break down that protein. So we have this protein substrate, and what happens is we could actually bind what we would call ubiquitin to that protein, which is a signal for a very important enzyme called the proteasome, which would actually break down proteins with ubiquitin chains on them. And so this is actually an energy demanding process, so it takes up ATP, but eventually what we do is we can break apart that protein into its amino acids, or even dipeptide, tripeptide chains, or unique chains of peptides for antigen presentation. So this is actually could be involved in our immune system, and if we have invaders of a specific type, we might break down the proteins associated with that organism and then present it to our immune cells. Now, also, we could just simply break down the protein entirely, collect the amino acids, recycle the amino acids. So that's the ubiquitin proteasome pathway. So that's breaking down proteins.